Assalamualaikum ladies and gentlemen, welcome to new video. Today's topic of our video is protein metabolism, and we will discuss urea cycle, and link of slides on this topic for your preparation is given in the description below. Thank you. Let's start. Bismillah ar-Rahman ar-Rahim. Today we will discuss the urea cycle. Urea is the major disposal form of ammonia groups derived from amino acids and accounts for about 90% of nitrogen containing components of urine. One nitrogen of urea molecule is supplied by free ammonia and in other nitrogen by aspartate. The carbon and oxygen of urea are derived from carbon dioxide. Urea is produced by the liver and then is transported in the blood to the kidney for excretion in the urine. All the nitrogen containing compounds are toxic for human life. Nitrogen convert in human body into urea, and this urea is excreted from the body in urine. Urea is a major disposal form of amino groups. Reactions of cycle. There are total 5 chemical reactions of urea cycle. The first 2 reactions leading to the synthesis of urea occur in the mitochondrial matrix, whereas the remaining cycles enzymes are located into the cytosol. Therefore, the remaining 3 reactions of urea cycle occur into cytosol of the cell. In the diagrammatical presentation of urea cycle, carbamyl phosphate is first formed by the action of carbon dioxide and ammonia and utilize ATP for this reaction. Carbamyl phosphate is converted into citrulline. Citrulline is converted into arginosuccinate. Arginosuccinate is in arginine and ornithine, and by the hydrolysis of arginine, urea is formed. And this is a complete cycle of urea which operate inside the body. Now we will discuss each reactions individually. In first, carbamyl phosphate synthetase 1 is driven by cleavage of 2 molecules of ATP. Ammonia incorporated into carbamyl phosphate is provided primarily by the oxidative deamination of glutamate by mitochondrial glutamate dehydrogenase. Ultimately, the nitrogen atom derived from ammonia becomes one of the nitrogen of urea. Carbamyl phosphate synthetase 1 requires N-acetyl glutamate as a positive allosteric activator. Carbamyl phosphate synthetase 2 participates in the biosynthesis of pyrimidines. The second reaction is the formation of citrulline. The carbamyl portion of carbamyl phosphate is transferred to ornithine by ornithine trans-carboxylase, as the high energy phosphate is released as inorganic phosphate. The reaction product citrulline is transported to the cytosol. Ornithine and citrulline are basic amino acids. that participate in the urea cycle. Moving across the inner mitochondrial membrane via a co-transporter, they are not incorporated into cellular proteins because there are no codons for these amino acids. Ornithine is regenerated with each turn of the urea cycle, much in the same way that oxaloacetate is regenerated by the reactions of the citric acid cycle. The third reaction is the synthesis of arginosuccinate. Arginosuccinate Arginosuccinate synthetase combines citrulline with aspartate to form arginosuccinate. The alpha amino group of aspartate provides the second nitrogen that is ultimately incorporated into urea. The formation of arginosuccinate is driven by the cleavage of ATP to adenosine monophosphate and pyrophosphate. This is the third and final molecule of ATP consumed in the formation of urea. Syntheses are collected in the form of ammonia and aspartate. The fourth reaction is the cleavage of arginosuccinate. Arginosuccinate is cleaved by arginosuccinate lipase to yield arginine and fumarate. The arginine formed by the reaction serves as the immediate precursor of urea. Immediate precursor of urea, fumarate produces in the urea cycle is hydrated to malate, providing a link with several metabolic pathways. And the final stage is the cleavage of arginine to ornithine and urea. Arginase, hydrolase, arginine to ornithine, and urea is virtually exclusive cleave arginine, thereby synthesizing urea, whereas other tissues such as the kidney can synthesize arginine by these reactions. Fate of urea. Urea diffuses from the liver and is transported in the blood to the kidneys, where it is filtered and excreted in the urine. A portion of the urea diffuses from the blood into the intestine and cleaved to carbon dioxide and ammonia by bacterial urease. The ammonia is partially lost in the feces and is partially reabsorbed in the blood. In patients with kidney failure, plasma urea levels are elevated, promoting a greater transfer of urea from blood into the gut. The interstitial action of urease on this urea becomes a clinically important source of ammonia, contributing to the hyperammonemia after seen in these patients. Oral administration of antibiotics reduces the number of interstitial bacterial responsible for this ammonia production. Regulation of urea cycle. N-acetylglutamate is an essential activator for CPS1 enzyme, the rate limiting step in the urea cycle. It increases the affinity of CPS1 enzyme for ATP and N-acetylglutamate is synthesized from acetyl coenzyme A and glutamate by acetylglutamate synthetase in a reaction for which arginine is an activator. The cycle also regulated by the substrate availability in short-term regulation and enzyme introduction as a long-term regulation. Thank you very much.
Thank you for watching our video. Please hit the like button, subscribe to our channel, follow us on Instagram for upcoming new videos.